everyone. Welcome to OsteoBites. Thanks so much for making the time to join us today. My name is Christina Iptoma, and I am mom to OsteoAngel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBites, we are talking with Dr. Alice Soragni about osteosarcoma organoid models, the potential and limitations of personalized avatars. Thank you so much, Dr. Soragni, for joining us on OsteoBites today. We are so excited to hear your presentation. And thank you to our panelist, Walker, for joining us today. Walker is an osteo warrior, and he is also Vice President of our MIB Agents Junior Advisory Board. A little bit more about our guest today. Dr. Alice Soragni is an assistant professor in the David Geffen School of Medicine at UCLA, and she's also a member of the Jonsson Comprehensive Cancer Center and the UCLA Molecular Biology Institute. She has a Master of Science cum laude from the University of Bologna, Italy, and a PhD from the ETH of Zuerich, Switzerland. Her laboratory in the Department of Orthopaedic Surgery at UCLA couples basic research into mechanisms of disease to the development of novel therapeutic strategies. Her expertise lies in the development of tumor organoid models to investigate the biology of rare tumors and to perform screenings for functional precision medicine applications. Welcome, Dr. Soragni, and welcome to everyone joining us today. Please feel free to add your questions for Dr. Soragni to the Q&A. And I just have to say, I had the pleasure of meeting Dr. Soragni like pre-pandemic. So you're actually like one of my sharpest pre-pandemic memories, because I think I was stalking you at a medical conference in San Francisco, like the week before everything started shutting down, when it was that awkward phase where there was tons of hand sanitizer around, but no one was wearing masks.
Yep.
And we were at this like very large medical conference. So that was like one of my kind of like, carefree, like pre-pandemic memories. It was so nice getting the chance to meet you. So excited to have you as a guest today. Thank you. Before we get started, I have some announcements and reminders. Factor, our annual osteosarcoma conference, is just 3 weeks away. We are going to sell out this year in Atlanta. We have about 10 seats left, just given the capacity of our conference space. So there's still time to register. For our scientific sessions, there are 6 panels. You can see what those are on our website, one of which is preclinical models, which will be moderated by Jason Eustein from Emory University. So if you're interested in our discussion today, there's going to be more content on this topic at Factor. And this year, we are looking forward to having 3 breakout discussion groups. So in addition to the presentations on our scientific panels, there really is space for everyone to participate in the conversation and have an interactive discussion. So our discussion topics this year are on comparative oncology and how canine trials can expedite pediatric trials, on how computational biology can accelerate osteosarcoma discovery, and also on patient-powered research. And then we also are going to have the amazing Lori Krause, who leads our Healing Hearts Bereavement Grief workshops. She's going to be joining us at Factor to do a Healing Hearts special session live in person there. But she's also going to be available for drop-in grief counseling for our families. And if you can't make it to Factor, we are still doing the virtual Healing Hearts. It's now through July 5th on Wednesdays at 7 p.m. Eastern. And if you're interested in those, I'll put more information in the chat about how to get more info on those. Walker, do you want to go ahead and introduce yourself?
Hi, my name is Walker. I'm the Vice President of the Junior Advisory Board this year for MIB. I'm really excited to listen to the presentation today. So Dr. Soragni, you can take it from there.
Great. Thank you both so much. I'm really excited that we were able to make this happen. So perfect. All right. So today we will go over a little bit of what my lab has been doing to really leverage patient-derived models for research, to learn more about rare cancers, as well as for precision medicine. Let me see if this works. Okay, great. So the basic idea, of what we do in the lab is we like to work directly with patient samples, because that's, as close to the real tumor as it may be. And we want to find ways to take these models and be able to do large drug screenings to identify new vulnerabilities, new therapies, etc. So when you work in that optics, you don't have actually a ton of options on what to do with these cells. Typically, you cannot just take them and put them in 2D, like in a petri dish on plastic, because they don't grow like that unless you modify them extensively. You can take these cells, you can put them in a mouse, and you generate what's known as patient-derived xenograft. Now, there is some absolutely brilliant biology done that way. But for us, as we are looking at performing potentially large drug screenings, it's actually very challenging to use PDXs, both because it's very expensive, takes a very long time sometimes to form these tumors in mouse. And ultimately, what you need to do in order to get a large number of mice with tumors is you need to passage them. So you take one mouse, you develop a tumor, you take that out, you chop it off, you make more mice. And as you keep going through this process, a lot of things can happen that actually change that tumor in a way that may not be very representative of the patient tumor anymore. And some of the sarcomas we studies, you just cannot get, PDXs to grow at all. But the easiest thing you can do is take these cells directly from the operating room and put them in 3D and grow them ex vivo in this way. So you take them, you put them like in a protein gel that recapitulate basically the structure of the extracellular matrix in the patient. They actually typically like to grow this way. They grow really well with very high take rates. And you are able to maintain certain components of, cell-cell interaction and of, interaction of the cells with the microenvironment that are really physiological. And, as we and so many other people over the years have shown, these models are actually really good at recapitulating patient responses. And so they are an ideal model for this type of drug discovery. When we started this work, the major limitation was really technical. Although in principle, this is a great idea. You can take the cells, grow them in 3D and perform screenings. Technically, there were very big limitations to doing this effectively in the lab. And these limitations have to do with the way that people would conventionally grow the cells in 3D. Generally, people would take them, put them in Matrigel, which is one of these matrices we can use, and then maybe, either deposit a drop at the center of your well in a 96-well plate, cover the whole well, or sometimes people like to use matrix-free, like U-bottom plates. This is a plate, the well looks like it has a U-bottom, and the cells kind of just aggregate at the center of that. What all of these methods have in common is they do use the center of the well, which really makes it complicated then to add to use liquid handlers or robotics to perform large, robust screening. So the way in which we went about this is very simple. Instead of using the center of our well, we grow the cells in what we like to call mini rings at the periphery of the well. So we deposit the cells with Matrigel at the edge, basically. And as you can see, this is one well of a 96-well plate. You can get your cells happily growing there, and your center is completely empty. And in this manner, you can use existing, liquid handler, any type of automation you want. In this, typically go to the dead center of the well. So you can go in there, you can remove the media, you can feed the cells, you can add drug treatments, etc., in a very facile manner and without ever touching or perturbing your cells in any way. So the way our overall general pipeline works is fairly simple. We get cells from any type of surgical procedure, including biopsies. And then we dissociate these into, single cell or small cell suspension, and we generate our rings the same day of surgery. Then we let this grow, establish, move around, get comfortable in culture over 3 days. And then after that, we perform through 2 drug treatments, 24 hours apart. 24 hours after our last drug treatment, we then perform a final endpoint assay. And in addition to the endpoint assay, we are also monitoring and imaging cells day after day after day. And we now have a nice machine learning-based pipeline to quantify their growth in culture over time. So why are we trying to do these things so fast, right? And so the idea is to try to apply this concept of functional precision medicine and going from surgery to a platform like the one we developed to identifying a therapy that could be effective for that particular patient within a timeline that is clinically relevant and therapeutically actionable.
Dr. Soragni, can I ask a super basic question? So it's like a terminology thing, because, we hear a lot about organoids or 3D models or sarcospheres. And so I'm assuming they're not all created equal, or they're just different methodologies for creating them. Can you just talk about that a little bit? I mean, is it kind of like with PDX where there's just different ways of, there's an orthotopic model?
Right. It's a great question, Christina. It's actually a combination, right? A little bit is different methods and a little bit is semantics. And another bit is the field is kind of developing, and we're coming up with these different names and ways to call things. And we don't necessarily agree with each other. I can tell you, for me, an organoid, and I will make that point later when I show you how the sarcoma organoids look like. An organoid, by definition, it's something that looks like the tissue it came from. And so if the organoids you develop have the same characteristics and they look like the tumor they came from, to me, that's appropriate to be called an organoid. These are typically grown in an extracellular matrix. Sometimes when people talk about sarcospheres or spheroids, they tend to talk about 3D culture that's matrix-free. And so this doesn't have that extracellular component. It has, for example, this U-bottom plate where basically the cells just don't have the ability, because the plate is just a non-treated plastic, they have no ability to attach to it. And so they can do nothing else but attach to each other if you wish. So it's a slightly different system, and the biology of it is a little bit different as well. But again, it's a spectrum, it's a continuum, it's confusing. And I think as a community, we need to do a little bit better to, agree on the terms. Let's put it this way.
Thanks.
Of course. And so as we were mentioning, right, so precision medicine, everyone is familiar with the concept of genomic precision medicine, right, where you sequence a tumor. You find an alteration that's specifically, in the tumor and not in the normal cell, and you target that with a drug that was developed for that specific purpose. The concept is brilliant, and the application hasn't worked as great, as we wish it did. The first thing is, these are some of the results of the NCI match trial. There isn't a lot of patients which are found to have what we call the targetable alteration, right? Overall, we're about, in the 32, sometimes if you combine multiple drugs, 40% of patients, but it's more like, 20 to 30 on a typical case. In addition to that, when you couple this intervention, you can see the purple bars are the responders. So there is actually very, very low response rates. But even if these patients were cured, you're still left with 70% of patients for which you don't have a targetable, alteration. So the idea here is that we don't need to know much about the tumor. You don't need to know if there is a targetable alteration, because we're simply taking the tumor and test out everything we can in order to find something that works. And we do this both for chemotherapy, which is, not going anywhere, not going away anytime soon, as well as for targeted therapies. And as I mentioned, some targeted therapies have a biomarker, but as you saw, there is still a lot of people that have the biomarker and don't respond. And there also can be some patients that do not have the biomarker, but may be responding to these drugs. And so we could identify these patients. The majority of targeted therapies do not actually have a biomarker at all. So we don't know how to find which patient could be responding to which therapy. And so the idea is, again, we can just test it out, right? And we can then use this data together with sequencing to develop those biomarkers that are sorely missed, or to improve on existing biomarkers, if you wish. So my lab does focus on rare cancers. Osteosarcomas, obviously, as well as all sarcomas, qualify as rare cancers. And I think the thing about rare cancer that I do want to mention, and I know this community knows this very well, but rare individually, it's one thing, but realistically, depending on the definition you use, up to a quarter of all cancer diagnoses are actually rare cancers. And so that's a lot of people with a cancer that we may know very little about. We may not have standard of care. Definitely challenging to run clinical trials in a timely manner. And so I think there is a lot of need, for rare cancers to find better ways to model them, identify interventions, etc. So our sarcoma pipeline is just, as I showed you at the beginning, we get all types of tumors and sarcomas, from primary, local, metastasis. We get tissue through biopsies, we get tissue through resection, and then we just kind of go through our pipeline. So we digest the tissue, develop our organoids, and then do drug screening, characterize their histopathology, as well as perform sequencing. So we have a preprint out, which is the result of the first 127 consecutively enrolled sarcoma patients. We are now up to, I believe, about 160 patients enrolled. And this first study talks about 200 samples or so. We are now probably close to 300. And so UCLA is a large sarcoma center. We get a lot of sarcoma patients. And as you well know, sarcomas include osteosarcomas, Ewing sarcomas, a few others, which are more common in the pediatric and young adult population. So about half of our samples is from younger patients, skew slightly male, as the biology is, and we have a majority white population. And you can break this down for the different histologies we looked at. And in terms of sample characteristics, as I mentioned, it's close to 200 samples we looked at for this first paper. We are a referral center, so the majority of the patients that get to us with advanced disease. So we have a lot of metastatic and recurrent cases. And we have a lot of samples that have seen previous lines of therapies, right? It's literally almost the minority is samples that are therapy naive. A proportion has seen radiation, and the majority has seen other types of systemic therapy. We screened about 50% so far of these samples, so about 300 plates or 29,000 wells we looked at. And our take rate, it's actually 93% or so. And as I mentioned, when you break it down by characteristic, you can see osteosarcoma is our largest sample population here. Majority are advanced lesions, which I've seen a lot of therapies before. And we can really pick up, specific trends. For example, radiation, we see a lot of radiation in samples that are soft tissue sarcomas, right, which tend to be treated by radiation, etc. So we can really look at the detail in a granular manner. Now, how do organoids look like? They look as diverse as sarcomas are. Sarcomas are very heterogeneous. And there are over 100 and I believe 20 or 30 by now types of sarcoma described. They look very different, and the organoids look very different as well. But the idea is, can we capture the basic characteristics of the tumor of origin, right? And so that's why we keep the organoid nomenclature. And so as you can see, we have very different tumor types. And what's really important for sarcoma is that some sarcomas are not very cellular. They are cells interdispersed in a dense matrix, right? And that's what we want our models to look like. We don't want to just develop, a ball of cells. We want to have them grow just as they do in the patient. And as I mentioned, we have a pipeline that uses imaging to understand how they're growing in culture. And so we can monitor that as well over time. Then we do drug screening, right? So we have these organoids. We can now perform drug screening. We can do all sorts of different types of therapy. We can test, single drugs. We can test 3 drug combinations. We can test two drug combinations. We can kind of do it all, and we can pick up, very distinct types of responses ex vivo. The other thing we can do now, because we have tested so many samples, is, okay, we can look at the samples individually, but we can also look at the samples in the context of all other samples we screen, right? So we can do this for osteosarcoma. These are all our osteosarcoma samples. We have screened them for a number of different drugs. This is just a small selection. And so we can kind of see how, well do they do compared to all the other osteosarcomas. And I think what you can pick up from these is that yellow means not responding, and red means, strong sensitivity. So there is a spectrum, right? Some patients seem to respond more to one versus the other. What I think is important is we never find, we tend to, it's very rare that we find a sample that is sensitive to everything, right? Just indiscriminately sensitive. We tend to really pick up individualized responses here. And as I said, as we take these cases and we look at them in the context of all these sarcomas, we can also try to start to tease apart what potential trends we see, right? For example, do we see any characteristic that correlates with age? And we do this, right? And we can see a few trends which are, we don't understand necessarily the biology behind it yet, but we can, for example, see that for a specific drug, this is cediranib, there is the adult sarcomas respond much less than the younger population. Or vice versa. This is a JAK inhibitor for which, pediatric and AYA tumors tend to be more resistant than adult tumors. So these are just interesting trends that we are kind of starting to pick up from these very large data sets. And the other is, can we see any correlates by sarcoma subtypes? So can we pick up things that osteosarcomas respond better than the average of all the other sarcomas we tested? The answer is yes. We see a number of trends. We can see, for example, ceralasertib, cabozantinib, everolimus. Some of these drugs, I'm sure are familiar to those of you that know osteosarcoma and therapies, etc. For example, ceralasertib is now in trials together with olaparib for therapy of osteosarcoma. And we can see that on average, osteosarcomas tend to respond more to this drug than all other sarcomas we test. And while and I'll show you a little bit more of this in a second, we also pick up the opposite trend. There are things, for example, to which osteosarcoma responds less than other sarcomas. Just to show you, we have a little bit of all these different possibilities in the data set. But as I said, the teal color here is osteosarcoma. You can pick up trends where you see blocks of osteosarcomas being, for example, more sensitive, in this case to sorafenib, everolimus, than other types of sarcomas. But even as you do that, there are always individual osteosarcomas that actually do not follow that pattern, right? Which is kind of, again, speaking to the idea that a personalized approach may be better to kind of pick up these outliers along the way. And I just want to have a quick example of how sometimes sequencing and our testing do match. This is on a spindle cell tumor. It's one case of a very young patient with a very aggressive, undifferentiated spindle tumor, which was found by sequencing to have a mutation in the PIK3C alpha gene that is conferring sensitivity to alpelisib. This is a hotspot mutation. It's one of the most common mutations found in this gene. And just expressing this mutant in the cell line confers sensitivity to different molecules, including the PI3K inhibitor alpelisib. When we go and look at the patient's sensitivities, we can clearly see that every PI3K inhibitor we tested was actually showing up as a drug that is able to kill these cells. And when we put this in the context of all the other sarcomas we test, this patient did very well. It's the second most sensitive sample we have ever picked up for alpelisib. And as I said, we sequence the genome and we can find this mutation there. Very obviously, we had a second patient coming in shortly after, which is a 134, is an MPNST. Was found on tempus sequencing to have the same mutation on the primary tumor. This was a recurrence just a few months after. We get this sample, we screen it, and we find no response, right? And we're like, what's happening here? Now, very interestingly, we went back and we sequenced the recurrence, and there is no alteration anymore. So that PI3K mutation, for some reason, is not here anymore. And we're investigating what happened to the tumor and what type of evolution is going on. But I think what's the most interesting story here is that this top responder to alpelisib is an osteosarcoma, which also doesn't have that biomarker. And there wouldn't be any, I think, reason to suspect that this tumor would be responding so strongly, right? And so again, now we have this data, we have the sequencing, and we can try to come up with better biomarkers here. One thing I didn't mention, but I kind of alluded to, is we go very quickly from surgery through the pipeline. What that means is that we actually do not grow these cells ex vivo. We don't expand them. We don't turn them into cell lines. So we're very limited in the things we do by the number of cells we get. This is by design, but it is a limitation, right? And so we need to kind of take this into account as we design our screenings. We tend to prioritize FDA-approved therapies for a cancer, first for the indication and then for cancer indication, as well as NCCN recommended regimens and therapies in trials. And these, molecules which are of high interest, we perform small dose concentration type studies. And then whichever extra cells we have, we can do larger than primary screenings. These are more of discovery screenings, so single concentration where we probe more broadly to try to find potential sensitivities. And then we always can go back to cells we freeze at the time of surgery to validate the results afterwards. We can make do with very small samples. This is one example of a chondroblastic osteosarcoma diagnostic biopsy. So we got lucky here. We could intercept that biopsy and get a little piece of it. And so this is how the tumor looks like in the patient, and these are the models we developed. And again, I kind of want to get your attention to the fact that sometimes sarcomas have these very scattered cells, and we really want to be able to capture that. We could test about 80 different drugs on this sample. And the one thing that jumped at us was a sensitivity to platinum therapy. Now, any patient who's diagnosed with osteosarcoma goes on to receive, or virtually all of them, MAP, which is a combination therapy that includes platinum, which is what happened to these patients as well. And a couple of months down the line, at the time of resection, kind of have to put a frame here because you can see there is nothing left in this tumor. This was entirely necrotic, over 99% necrotic, which, as many of you know, it's a really good indicator that, this kid is likely to do very well long-term. So very good efficacy of MAP. And we could kind of pick up on these very early on, right? We are now trying to do more and more of these. It's very challenging to capture that diagnostic biopsy outside the context of a trial because, life happens and patient can be coming in at any time. So it's a little bit logistically challenging. But we have been able to capture a couple more where we actually tested the full MAP regimen on the organoids. And we are seeing a really good correlation whereby, a sample, for example, where the organoids are dying nicely, are dying nicely on MAP, there is actually a very high level of necrosis at time of resection. And vice versa, sample that is not dying as well on MAP was correlated with a lower level of necrosis. And so we're seeing a good trend here, and we really want to try to grow these numbers. And so we are trying to open a trial now where we kind of, put a framework around these and try to have an easier time accessing a little bit of these diagnostic biopsies to really prove what's the prognostic ability of this type of assays. And now, again, I know it's not surprising to anyone that these patients, get MAP, but I still think there is value in knowing what type of response to expect, right? We are also trying to look at other types of therapies. Oh, there you go. As I said, everyone gets MAP as first-line therapy pretty much. That's not super surprising. But already, second-line therapies for osteosarcoma, disease that's relapsed, refractory, metastatic, you have a lot of different regimens, which are NCCN recommended, and not necessarily preponderance of evidence as to which way to go. And so the idea is, can we provide this additional information to help the oncologists kind of pick and choose which one would be the better treatment for each patient?
Soragni, you mentioned that you just need a really small amount of tissue. So are you able to use like a core needle biopsy amount of tissue?
Yeah, this is what these are. Actually, these are core needle biopsies. So it's, an extra core, an extra couple of cores. If we, the way in which we're trying to build the trial also is, to limit the number of cores. We don't need a lot of it. And it may fail in some cases, right? It kind of depends on the tumor, how cellular it is, which area is being biopsied. So it's not going to succeed in every single time. But we have had a decent success with biopsies. And so the hope is that just having an extra core or so could allow us to test, this one regimen, for example. So yeah, the idea is we try to minimize, we don't want to, add any discomfort to the patient on any additional procedure. This is really like, can we get a little speckle of what goes to pathology and do a little bit of testing. And so we are trying to look at also correlation with other types of regimens. Again, outside the context of a trial, this is very challenging. What we are trying to do is we are taking patients that just after surgery went on a regimen that we actually tested on the organoids from that same surgery. So, we don't want to say, oh, we tested this therapy on organoids, and then a year later, the patient got that therapy, because in the meantime, maybe the patient got other 5 therapies and the tumor may have changed, and, we cannot really make a correlation that way. But, if we take the patient at that moment in time and the therapy that is being given right away, immediately after surgery, we can try to see how well the organoids predict the response. So we have a small number of patients, mostly osteosarcoma, actually, where we were able to do that. The way in which we're trying to quantify this is by looking at time to next therapy as a real life measure of efficacy. What happens here is that as these are advanced diseases, and as the patient progress, they tend to stay less and less time on the next line of therapy, just because the tumor is getting more aggressive and developing, resistances, changes to the genome and whatnot. So if we identify a regimen on which the patient can stay actually longer than the previous one, that tells you there is a signal of efficacy there. And so where we see organoids dying nicely on a regimen, we can also see how the patient actually tended to stay longer, maybe over twice as long as the previous regimen. And vice versa, which I think is it's another really important piece of the puzzle. When the organoids were doing really okay on that drug, the patient also actually very quickly moved on to the next therapy. Again, telling you the patient probably progressed really rapidly and had to move on and try whatever comes next, right? And so we're seeing a decent correlation here. And part of the trial we're trying to build is also going to be on very advanced patients, trying to see if we can identify, regimens for that patient population. And so trying to get more numbers, again, to understand what the prognostic ability of these assays are. The other thing we're looking at is what types of drugs are coming out of this assay. Would that be actionable at all, right? Because if we're screening a bunch of drugs which are not available to patients, and we try to think about implementing this type of assays for precision medicine, there is really not much we can do. And so we looked at whether what type of drugs are coming out. We look at the top 5 drugs for each patient we screen, and we only consider that a hit if the patient is also within the top 10% of responders. And we do this to try to avoid, for example, drugs that are, just poorly active on very resistant samples. So we have this way to calibrate. And the drugs that are coming out of the screenings are approved drugs. Are they approved for the same cancer indication we found it? Sometimes the majority is yellow, which tells you they're actually approved for another cancer indication. But as long as the drug would be accessible, whether on or off label, we consider that as an actionable result. So when you put it all together, all the patients we screen, we can find at least one FDA-approved or NCCN recommended regimen or a drug in trial for at least 58% of all the patients we screen. So this is a fairly large number, particularly if you compare it to the 30 or 40% of patients found to have actionable, information by genomics. So this information, could be actionable as long as, it goes to trial and it's proven that you can predict patient response, of course. And, I think when you look at osteosarcoma is in this optic, it's also interesting because we can see a small percentage of cases responding to each and every approved regimen. But there are some responses, at least for second-line therapy. I think what's very interesting here is that, although I showed you at least one case where we saw a response to MAP that was matched clinically, that response was not within the top 5 most effective drugs for that patient. And this is nothing but food for thought. But the idea is, maybe in a far future, if these type of things can be, proven clinically and whatnot, and they pan out, you may have the ability to give, a different type of therapy, right? But the kid did respond to MAP, so that's fantastic. We have a little bit of data now comparing this to PDXs. I don't want to go into detail, but we see, despite the fact that the timelines are so different because we are done in a week, and PDXs, anywhere between 6 months to a year, we still see some hint of correlation, which honestly is better than I even expected to see. So we are doing this work as well. But, what are the roadblocks ahead? So there are regulatory issues, on how to take this type of assays forward clinically. There are, some issues we're going through to design trials that are meaningful and can give us the type of data we need to prove or disprove whether we can demonstrate clinical efficacy. Access to drugs is a huge problem. We don't have the time to go into this today, but definitely an issue. But the bigger issue here is, and really the last thing I want to mention is tumor heterogeneity and sampling bias. This approach, like any approach where you take a chunk of tumor, you are biased towards that piece of tumor you are sampling, right? This is true when you're doing, even when you're looking at a bigger tumor, so biopsies within a primary tumor. And it's even more true for when you're looking at different metastases. I'm just going to show you a quick example or two on how this looks like. This is one case of a very advanced metastatic osteosarcoma. And here you can see one of the lung nodules in the patient. This is our models, and I think you can appreciate they're really hard to distinguish. We got 2 samples from 2 individual lung metastasectomies from this patient, which were taken about 6 months apart. I think the patient went through about 6 lines of therapy before the first surgery, and then an additional 2 to 3 between surgeries. When we go and look at these tumors, what was quite astounding is that pretty much beside our positive control here, anything that had a signal of activity on one metastasis did not have any activity on the other, and vice versa. So it's almost like looking at 2 different tumors when the disease is so advanced.
Is the patient on the same therapy in between the surgeries?
Was through between surgeries. I think the patient went through at least a couple of lines of therapies, of different therapies. They not related to what we tested. We're just saying, this is just a completely not looking at therapy, not looking at response. This is just looking at, signals of activity. And the idea is just that if you try to implement this type of assay for very advanced patients where there is a lot of disease, and you're sampling just one location, you may be finding something that's effective, but it may be effective only for that tumor. It may not be effective on these other, 3 or 4 metastases that are there, or any other type of occult disease, etc. And so when you look at guiding therapy, you need to be mindful. Because again, you can be doing a very good job at eliminating that clone or that proportion of cells, but everything else may look very different. So we're trying to go about this by trying to learn more about this heterogeneity, right? What can we figure out? And can we understand the biology a little bit better to come up with better ways to do this? And so there are patients now where we are trying to go through, which we can follow through the progression of disease, and try to capture, the biopsy at the very beginning, capture the sample from the resection, capture different metastases as different surgeries may be happening. This case in particular is a patient presenting with metastatic osteosarcoma. So about a quarter or so patient that go from, being healthy to having metastatic disease right away. And we can try to take these different metastases and we can see how they grow in the patient. We can compare to how they grow ex vivo, and we can perform large drug screening. This is just a little piece of, a much longer chart where we are looking at 3 different samples, the biopsy and 2 of the lung metastasectomies. And there isn't really anything much here. I just want to give you the idea that we see very interesting patterns. We see, for example, sensitivities that arise over time. Again, the tumor is changing. We see sensitivities that go away. And we see individual, sensitivities for specific metastases, right? A little bit what I told you, that one has to be a little bit careful on how to go about this. And we are doing a lot of work now in collaboration with the Boutros Lab to deeply sequence these and look at what's happening at a subclonal level. And, we are doing subclonal reconstruction. And I think, as you can see, tumors are changing as they go to metastasis. But also the other thing I want to point out here is that we are doing the sequencing also on the organoid. And this is looking at CNA clonal reconstitution. And you can see that the biopsy and the organoids from the biopsy really have the same populations of cells, even within the same percentages, more or less. Similar here for this organoid of a metastasis, or here for these organoids and metastases, different metastases. As the tumor is changing, we're able to capture those changes to a very high degree of fidelity, which again, it's really important for performing this type of drug screening or thinking about precision medicine, if you wish. And we are, the other thing, but I'm not sure how I'm doing on time. I'm going to ask you because I've been talking about, we are trying to develop new technologies to look at heterogeneity better. And one of the most exciting things we have, which I'm just going to give you, a little snippet about, is this new platform whereby instead of seeding those rings of cells I showed you at the beginning by hand, we now can print the cells. We can use a printer. We can print the cells as mini rings. We can print them as mini squares, as any type of shape you can think of. And because we are printing them, we can make these very thin layers of matrigel that now allow us to use much better and interesting type of imaging technologies. And this is a collaboration we have with the Title Lab at UCLA, where we are using this type of imaging, which is called high-speed live cell interferometry. And what it does, very simple, it measures the weight of cells. So you can measure the weight of individual organoids here. And let me just show you, and this is just the printing, but let me just show you how it looks like. So we can literally go in and see the individual organoids and see how heavy they are and how they move around, how they fuse sometimes, and all types of things they do in culture. And the good thing here is that this is completely label-free, is spatially resolved. And so we can kind of go out and measure how each individual organoid is responding to therapy. And we know which ones are not responding. And so we have now the ability to go back and kind of pluck these out and analyze them better to understand heterogeneity further. And what I showed you was cell lines. That was our proof of principle. And we have a paper coming out next week in Nature Communications showing that, but we can also do this in patient and in sarcoma. And so we're starting to do this larger screening, measuring at weights of organoids in sarcoma as well, which I think is going to be very exciting because, again, we can really kind of understand heterogeneity better with this type of technologies. We have other things we are developing. For example, we have a vasculature model where we're trying to add vasculature to the culture. We have a mini liver insert where we are placing a liver just where we need it to, for example, metabolize prodrugs that need, that type of metabolism to happen, to be active. We have an hypoxia platform. We have a chemoradiation platform that's going to allow us to do very interesting things in sarcoma. So it's, these are just all different things that we can do, as well as an immuno-oncology platform we are looking at to try to do immuno-oncology drug testing. And so this is our system. It's fairly simple, it's robust, it's fully automated. We don't need a lot of cells. We don't need any passaging. We can passage, we do passage. We have, we have projects like that, but we don't need to do it if we don't want to. And we can develop these personalized models, which we can use for personalized prediction, for markers, to do diagnostics, landscape analysis, and really understand heterogeneity better. And that is it for me. I just kind of want to thank all the amazing magical people in my lab that make all of this happen. Particularly what I showed you today was work by Wynn, Peyton, Hamad, and Bowen. And very thankful to our funders for the support and all of our clinical collaborators at UCLA and beyond. And thank you for listening. Happy to take any questions.
Thank you so much. And the preprint that Dr. Soragni referred to, I put the link to that in the chat, and we'll also send it out. If you don't have time to copy it now, we'll send it out when we send the link to the recording so that you can have that. But a lot of deeper information in there. So definitely great to check that out. Feel free to put your questions in the Q&A. And Walker, do you want to go ahead and take that first one?
Yeah. The first question is, after freezing the organoids, what is the success rate of reestablishing the organoid after thawing? In these successfully reestablished organoids after thawing, are there differences in the tumor-associated cells?
Yeah, that's a great question. We have not looked at it systematically, but just, anecdotally, typically, The absolute majority of sarcomas we have frozen, we were able to revive successfully. You know, there are tumors which are incredibly necrotic, for which we don't have a lot of like viable cells to start with, and maybe more challenging, but overall it works quite nicely. And in terms of changes, we have done for a few samples, we have screened before and rescreened broadly after thawing, as well as done targeted validation. So we screen broadly on the first sample, and then on the second one, we do just test a few separate compounds, and we see very good correlation. So far, we haven't had any case where, say you're freezing, you're losing a whole population of cells that didn't survive freezing, and then you see a different response. We didn't see that yet.
Makes sense. Thank you.
And I think we have another question that just came in from Jeff.
Oh, yeah. The next question is, are you interested in generating the RNA sequence data for the patient tumors and investigating how gene expression is associated with response to the targeted therapies tested?
Absolutely. We are, so bulk RNA sequencing, it's the easy part. We have generated data for not all of them yet, but maybe about 20-ish samples, and we're going through the queue. We have RNA-seq, samples. We have samples for RNA-seq for every case. Single-cell RNA case, sequencing a little bit more challenging, a little bit more expensive. We have done a small pilot study doing single-cell RNA-seq on pediatric sarcoma funded by the Alice and Monet Stan Foundation, but it is just a few samples. So it would be great to do this more broadly.
Dr. Soragni, I'm not sure that you covered it in your slides today, but I was in your in the paper. Something I thought was really interesting, and I'm not sure if I interpreted this correctly, but one of the things you noted is that for the samples that were heavily pre-treated, that they seem to have higher sensitivity to, which was kind of like counterintuitive to me, right? Because I would have thought that the heavily pre-treated would have developed so much resistance, but you noted that heavily pre-treated actually had higher sensitivity.
And it's absolutely. So I think maybe the slightly misleading point is that it's not that in general they tend to respond to more drugs, but there is a small number of compounds, and this is a few percent, like 2-3% of all the compounds we tested, where we see this very peculiar trend. And I completely agree with you. We're not really sure of what's happening. But there is a small number of drugs for which we see that heavily pre-treated samples are responding more. Now, here is the confounding that. Highly pre-treated samples in our cohort looks like metastatic osteosarcomas for the absolute, most part. And so there is a bias there of disease, basically, so type of sarcoma. And there is a bias of treatment paradigm, right? Again, the majority of the samples in that category tend to be younger patients, tend to be patients which are treated more aggressively. And so there are some confounders there, and it maybe we're just picking up a little bit of the biology of, some type of metastatic osteosarcoma or some such. So we will need more numbers. We try to address that in the preprint. There is this confounding effects. We need bigger numbers to really kind of tease apart what's happening. But some of these bias will be hard, will always be there, just for the nature of the disease and the heterogeneity and the type of tumors we get, if that makes sense.
Yeah. And, just kind of seeing this and kind of reading the paper, it kind of just made me rethink and revisit again the whole idea of precision medicine. Because I do think sometimes when we talk about that now, we still are kind of bucketing, right? Like we're kind of looking at like a subtype, of the disease, for example, versus truly individualized medicine. And kind of some of the things that you brought up were, there were a lot of things that affect sensitivity, like there's so many variables, right? It was like age, stage of disease, perhaps, but certainly like any pretreatment, right? So any therapies that patient might have been exposed to, whether that was systemic therapy or radiation therapy, at different points in time, and as the tumor changes. And so it does just kind of like if I'm a patient, like I would want this versus just saying, oh, I think, especially, I think because you had mentioned that you have some folks that respond without the biomarker, or they have the biomarker and then don't respond. So there isn't also like a direct correlation there. So just kind of thoughts on that. I mean, just when we talk about precision medicine, that really should be thinking more about really individualized medicine versus even this kind of bucketing by subtype.
And, there are some fantastic success stories, say larotrectinib, right? So there are very specific events like NTRK fusions, which have very good drugs, and we can get 90% of the patients having fantastic responses. But that is, unfortunately, the exception at this time. And so, yes, a lot of patients do have the biomarker and don't respond for the majority of the drugs, that people have been looking, the NCI has been looking at. The other thing which I think is super important is these changes over time, right? And so say that you have a biopsy and you get this type of testing or sequencing. And then, the patient goes on and receives traditional therapy and whatnot, many, many lines of therapies later, years later, the tumor recurs. What I think, and I want to stress all the time, and it's very important to me, because it is very scary looking at the biology. You cannot really take that all the information and think that the tumor is going to be exactly the same. And the things that may have been active back then may be active now. And so I think, and I think this is as true for sequencing as it is for us. And so any type of technology that takes a piece of the tumor, I think you have to be very mindful as to how the tumor is changing, particularly for things like osteosarcoma, which we know tend to have complex genomes and a lot of potential, changes happening over time. And so resampling the tumor at that time, I think maybe a more sensitive, approach than, 2 years ago, this tumor showed sensitivity by whichever means to these drugs, let's do this now, if that makes sense.
Mm-hmm. And I think you had also noted that one kind of benefit of doing this type of testing too is that you could kind of discern different response to even drugs within the same class. So that even though if you might have been directed to look at like I'm making something up right now, but like a PARP inhibitor or something, that even within that class, the samples might have responded differently to drugs within the class.
And so what that shows you is that what we are picking up on is how, let's call them dirty, some of these drugs are. So we think the drug does one thing. The drug, in a perfect ideal world, where there is nothing around the drug, but the one protein it's interacting with, which is what you study in vitro, certainly does that one thing. But in the context of the cells, any single drug tends to have multiple effects. And so I think that is what we're picking up on. Each and every drug, even though their main target is the same, may have very different secondary effects. And so that really is the type of things we are kind of picking up. And I think what was a little bit reassuring is we have this very large pathway analysis where we are taking drug responses we're taking drug responses, we're taking the drug, and we are looking at their primary and secondary targets. And then we are looking at what pathways are these primary and secondary targets impinging on. And are, so we can kind of pick up which pathways, not by drug, but by pathway, what pathways are sensitive to treatment in these sarcomas. And what that shows us is that although we may be picking up, different drugs for whichever reason, samples from the same patients tend to largely have similar pathways that are important for tumor response. And so, it may not be as drastically, complicated as I mentioned, but I still think heterogeneity is something we need to keep in mind, most definitely, yes.
And then, so one thing I can't ignore too is you had a chart that kind of showed, different therapies, the NCCN therapies map, of course, which really didn't seem to have like a lot of sensitivity.
No, it's more like it's not amongst the, so no patient had MAP as their most effective therapy. they may be responding to MAP, but they also were
was
responding to 5 other drugs way better than MAP.
Right. One of which I think, if I'm remembering correctly, Jim said minoxidil was kind of the winner in that horse race.
Well, for those samples, the one where we see a larger proportion of patients, responding effectively is salvage therapy, docetaxel, gemcitabine. We see a whole spectrum, right? So we see complete resistance, and we see a few patients which have for which that therapy was amongst the most effective. Correct. Did those patients get that therapy? Not necessarily, but we saw it as the most effective. Yeah.
Mm-hmm.
But the percentages are still small, and that really recapitulates what you see in trials, right? And so we see about 5% of patients responding to sorafenib everolimus. In trials, you get 2 responses out of 38 patients, so very similar rates.
So, so practically speaking, so I just have some questions about like you and interesting. So it looks like you did do a head-to-head on the PTX versus organoids. You had a slide on that, and so it looked like you found that they really did behave pretty similarly.
Surprising, honestly, to be completely honest, I was not expecting that. We have 2 or 3 patients now for which we have both, and we see a correlation. Do we see perfect, correlation? No. But you can see there is a trend that is still present despite the long time it took to generate PDXs. And so, there is, which is great, right? It's better than I would ever have expected.
Yeah. So can we maybe, oh gosh, we only have like 3 minutes left, but we do a quick pros and cons, organoids and PDX. So organoids, they're fast, right? Like you're showing like a week time frame between surgery and actually having kind of a test results. And then I'm imagine that cost-wise, it's also a lot less than.
Cost-wise, yeah, the difference is huge. Yeah.
Yeah. And then the take rate. So I think you had mentioned the take rate was like 95% in organoids.
So I think in average on the patient we have in the sample, in the paper is about 93%.
Yeah.
Now we are much higher. What happened with the paper at the very beginning is we were accepting samples from patients that were very recently irradiated. Turns out that those cells don't like to grow anymore. And so that kind of skewed us. Now we don't enroll a patient if radiation was received within a month from surgery. And so right now we have higher take rate. But yes, the take rate is much higher than PDXs. I cannot really give you a number for PDXs off the top of my mind, but particularly for bone tumors, those take rates are not.
Right. So so then let's talk about PDX. Like what are some of the advantages of PDX over organoid? Because we have, we talked about time, cost, take rate.
Right. And for other types of studies where you're not trying to screen broadly, you can propagate these cells, right? So you have the ability, for example, you can even take these PDX cells and then make organoids out of them and screen more broadly. So there are things you can do with the cells you get from PDXs. I think, the divide there is not as much in the biology as it is in the precision medicine angle. When you have to wait 6 months or more, and the patient may be going through additional therapy, I think you're kind of, then you're going to try to fight with the evolution of the tumor as you catch up on time. So then, but you asked me about the positive. The positive is, there is more cells around the tumor, which are, which can contribute to the way in which the tumor behaves. There is a circulation, so you can look at metabolism of drugs, in vivo. And you can give drugs that is, the whole reason why we're developing this liver system is because there are drugs we cannot use in the petri dish, right, without having a metabolism. And so you can definitely use, you can do eyes in a mouse, which right now we cannot really do well, for example. So ifosfamide and other types of drugs, you cannot do that.
All right. Thank you for that balanced. Right.
I'm trying.
I know. But you did. All right, great. Well, I put a little bit, I also just put a little bit of information. Thank you so much, Dr. Soragni. And we've kind of run out of time here. So we'll close up. But thank you so much for joining us and making it better for sarcoma patients. And more information on this and all OsteoBites you can find on our MIB Agents website, on our YouTube channel, and also at your favorite podcast place. And I also did just want to plug our MIB Agents Testing and Research Directory, which provides an overview of functional testing as well as molecular tumor profiling tests. It's been diligently refreshed by our osteoparent Jim Peterson, who's Dr. Brooke, and reviewed by experts like Dr. Soragni. And please join us next week on June 8th. We're going to be talking with Dr. Katie Janeway, and she's from Dana-Farber Cancer Institute, and Ben Zola from the Count Me In Project at the Broad Institute. And they're going to be talking about the Osteosarcoma Count Me In Project, what it is and how it correlates to the importance of patient-powered research and being more connected to the patient community. And by the way, they're also going to be, Count Me In also is going to be a factor, and we're having a full discussion as well around patient-powered research factors. So just another reason to come. You can find our OsteoBites lineup for the next few months on our website. And if you have any ideas for future topics that you'd like to hear about, just email us at events@mibagents.org. Thanks again, Dr. Soragni and Walker, and for all of you for spending an hour with us today. And we hope to see you back here next week on OsteoBites when we talk to Dr. Katie Janeway and Ben Zola about Count Me In. Thanks, everyone.